Hi Emma, could you start by explaining what chromatin is?
Of course. Chromatin is a complex of DNA and proteins, primarily histones, that together form the nucleosomal structure in eukaryotic cells. Histones are basic proteins that interact with DNA, wrapping it around themselves to create nucleosomes. Each nucleosome consists of 8 histones, and these units further coil to form structures called solenoids. This setup helps in compactly organizing the DNA within the cell nucleus.
I see. And chromatin exists in different forms, right?
Yes, exactly. During the cell's interphase comprising the G1, S, and G2 phases, chromatin appears in 2 main forms, euchromatin and heterochromatin. Euchromatin is loosely packed, making it transcriptionally active, which means it allows RNA polymerase to read the DNA and convert its information into RNA. On the other hand, heterochromatin is more densely packed, making it transcriptionally inactive.
So, euchromatin is associated with active gene expression and heterochromatin is not?
Precisely. Euchromatin is often lightly stained in microscopic images because it's less dense, while heterochromatin, being denser, appears darker. There are 2 types of heterochromatin as well: constitutive heterochromatin, which is permanently condensed and typically found at centromeres, and facultative heterochromatin, which can transition between being active and inactive based on cellular needs, such as the inactive X chromosome in female cells, also known as the Barr body.
Got it. Moving on, what about the cell cycle? How does that work?
The cell cycle is a series of regulated phases that a eukaryotic cell goes through from its formation to its next division. This cycle includes 4 main phases: G1, S, G2, and M, with each phase dedicated to specific cellular processes. G1 is for cell growth and preparation. S phase is when DNA replication occurs. G2 prepares for mitosis, and finally, M phase is where mitosis, or cell division, happens. Together, these phases ensure that each daughter cell receives an identical set of genetic information.
And I've heard of something called G0. What happens there?
Good question. After a certain number of divisions, many cells enter the G0 phase, where they cease dividing and enter a resting state with reduced metabolic activity. Some cells, like stem cells and immune cells, can re-enter the cell cycle from G0 when needed. However, cells in G0 can remain there for the rest of their lifespan, a state called cellular senescence or aging.
Fascinating. So what specifically occurs during the S phase?
The S phase is dedicated to DNA replication. Here, each chromosome duplicates, producing 2 identical chromatids. Additionally, the centrosomes, which help organize the mitotic spindle, are duplicated in preparation for cell division. Other organelles also start to replicate to support the creation of 2 new cells.
And after S phase, the cell enters G2, right? What happens there?
Correct. G2 is a preparatory phase before mitosis, focusing on further organelle replication and synthesizing proteins needed for mitosis. There's also a checkpoint to verify if all DNA has been replicated correctly before the cell proceeds to mitosis.
Speaking of mitosis, can you walk me through its stages?
Certainly. Mitosis has 4 main stages. In prophase, the nucleolus and nuclear membrane dissolve, and chromosomes start to condense. Microtubules from the centrosomes attach to the chromosomes at the centromeres, forming the mitotic spindle. In metaphase, chromosomes align at the cell's equatorial plane, forming the metaphase plate. During anaphase, the spindle fibers pull the chromatids apart to opposite poles of the cell. Finally, in telophase, chromosomes decondense, nuclear membranes reform, and the cell begins cytogenesis, where the cytoplasm divides, resulting in 2 genetically identical daughter cells.
How is the progress of mitosis measured?
We use something called the mitotic index, which measures the percentage of cells in mitosis at any given time. A higher mitotic index indicates a greater proportion of cells dividing, which is often observed in cancerous cells. This metric helps us assess cellular proliferation and the duration of each mitotic phase.
Interesting. And can mitosis be disrupted?
Yes, certain compounds known as spindle poisons or mitotic inhibitors can block mitosis. These include drugs like colchicine, which prevents microtubule formation, and taxanes, which prevent microtubule disassembly. Such compounds are used in cancer treatment because they inhibit rapid cell division.
Thank you. This has really helped me understand chromatin structure and the cell cycle.
Thank you for listening. If our podcast has been valuable to you, we would greatly appreciate it if you could leave us a 5-star rating and write a review on Spotify and Apple Podcasts, or subscribe, like, and share on our YouTube channel. Your support helps us greatly with discoverability. We hope to see you soon.